Hello, my name is Nathan Reyna, and I'm the PI of the Cell Biology Education Consortium. And my talk today is focused on how we use the CBEC to build undergraduate research infrastructures, and really my story of how I used it, and then my co-PI and others began to use these same ideas to build their research or build upon these ideas. And I'm at Ouachita Baptist University, a small private undergraduate college in southwest Arkansas. We have about 1,400 undergraduate students. About 20% of those students are biology majors. About 5 faculty, and I teach a full load. I teach 12 hours, usually, which means 2 classes. So 1 biology majors class, 1 non-majors class, and then I teach 2 labs, generally with the majors class. And so cell biology will run about 40 students with 2 labs of 20. And research isn't required at Ouachita in the sense that no one really cares if I get publications or I get grants. However, we want our faculty to work with students and engage students in research because we believe, and I believe, that this is really important for the learning and development of a student. And to sort of start off with my story is about 5 years ago, I was accepted or asked to be part of a statewide EPSCoR grant, or NSF EPSCoR grant. And about 30 faculty were involved. I was one of the only undergraduates faculty in the entire project, but they gave me a lot of money. They gave me almost $260,000 over 5 years, and they said, look, your role in this project is to help learn to see how neurons differentiate, maybe do some bioinformatics. There's all kinds of ideas that we think you should be doing, and we need undergraduates to be doing for this project. And I said, sure. But then I got to looking later on, a few years ago, at my initial sort of summary and research page that I submitted to be part of this program, and I couldn't even spell neuron. And so I had no idea what I was doing. I had no idea where I was going to go from there, but I said I would, and so I had to figure things out very quickly. And so we've been doing a lot of cures. And so I know there's a lot of people doing cures, and I thought, all right, here's our thing. We're going to do this cure. We're going to put it into the classroom. It's going to spawn off independent research, set all the great things associated with cures, and that's what we're going to do. The problem was that, for me, created a very top-heavy type of approach, where all my research was dependent on this class project. I did not have a very good foundation, and I had to find a better way to move forward. And for me, the way to do that was this concept of what we refer to as cell blocks. And what a cell block is, it's a video protocol, a written protocol, and classroom implementation strategy. So whatever technique we're interested in, we made a cell block. And my colleague Lori Hensley was already doing this. I used some of her cell blocks that she made just for her lab as a lab managing strategy for undergraduates to build out some of my projects and my ideas. And then what we noticed, which was really interesting, was her first videos that she created had over 25,000 views on YouTube in a short amount of time. And so it was really fun to start building these blocks and start seeing how they work. And what we did is we took blocks from all these different projects that she was working on, that I was working on, and we realized they could be mixed and matched because they all had a uniform foundation. And we wrote our grant for CBEC with the idea that other faculty and other students might have additional programs and protocols that they could add to it, and we could all mix and match it, and then we could pull from this pot, and we could create something unique that we were all individually interested in. And so rather than having a cure where we told others to all work on the same project, we kind of took a backwards approach where we created techniques and protocols that could be mixed and matched. But using that same theme and that same philosophy, you could do any research project that you're interested. And so an idea of how I use this in my classes is that each blue block that you see here represents a cell block. And these are all color-coordinated in the sense where every student in my lab or in my class will use this basic idea. And every cell block is created by students. And so, again, we have blocks being used in class, but we also have blocks that are being made by students through other types of projects. And in class, students then have different times they present. So they have to present why they're using this cell block. They present the results they have. And then they have to present, what am I going to do next? And this is where the uniqueness of the cell blocks come into play, because I then let my students decide which avenue, kind of a choose your own adventure, or which projects they want to do, and they have to justify it. And since the cell blocks are uniform and they're kind of modular based, it's not that difficult for me to grade a cell migration assay results as it is a cell differentiation assay results. And so students will keep doing this. They'll also have some presentations, come back to a normal or a uniform genomic analysis or Western blot analysis, depending on the semester or the day. And then we follow this up with presentations and data analysis. Well, if we're building a research infrastructure and I have money from this research grant, I got to also be able to do publications, and I got to be able to show to others that this works beyond just as a classroom model. And what was really exciting is we had our first publication coming out of the our work with the U87 or glioblastoma cell lines. It was published in collaboration with a friend at the medical school in a cancer research journal. Our cell differentiation work was first published in an undergraduate journal. A couple of the student authors in the undergraduate journal continued their project, continued what they were doing, and led to a very nice PLS1 paper that was published last year. Even our data analysis paper, as a result of COVID, we published a virtual lab and a virtual research lab that students could do using the Cancer CBioportal and doing cancer genomics. And so that turned into an education publication. And then the fact that we had that experience, we then went to a different researcher at our medical school, and my students gave context to some breast cancer data that he had already collected from patients. And so that publication is in review. And even our presentations, we created a Twitter poster concept where students could present their data, interact with others on Twitter, and that's now also in review. And so in a lot of ways, we've made a much more firm foundation to build our projects on. That not only are we doing the research that we're interested in the lab, but we're also doing really cool research in the classroom. And again, students are involved in everything. There's no more curtain. There's no more me trying to pull all these things out, show students how to do it. By having this idea of students building the cell blocks, students designing the questions, students developing the techniques as part of their own research, helps strengthen what I do in the classroom, which then helps strengthen independent research that turns into other projects and gives us more ideas that we have to do. And everything is sort of funneling through the idea of CBEC, or Cell Biology Education Consortium. And I can't say I do this alone, and I can't even act like I do this alone. I've been very lucky and very thankful to work with a lot of very good faculty, and these faculty are willing to collaborate with me in lots of different ways. And a lot of the times that we're interacting and we're collaborating is because of our ability to work in the lab, to take research in the lab and put it back into the classroom and move back and forth. And so there's a lot of research, there's a lot of faculty that need broader impacts, and they need to be able to show how their research will help others. They don't want to just come and show or talk to someone about their research. They want to show some true deliverables. And this model becomes a very true deliverable that other people to use. And so with that, I'd like to say thank you to everybody. I'm very appreciative of all the help, all the work. I also have to point out that Dr. Lori Hensley at Jacksonville State University is my co-PI, and again, none of this is done in a vacuum. It's done with her and her help. I would encourage anyone who's interested to contact us and join CBEC. We're more than happy to help people. We have funding to help students, to help faculty create more cell blocks. We have ways to show them how to create and put these things together. I'm also very thankful to ASCB for inviting me to talk, and I'm very honored and appreciative to be here today. Thank you very much.